I'm going to be talking about some work we've been doing for the last 2 or 3 years on bacterial contamination of the Indian River Lagoon. I want to acknowledge all the previous speakers who've set it up beautifully for me to take it from this point, so I don't have to explain what the Indian River Lagoon is or why we're interested in it. There are two major components to this study. The first one is looking at bacterial contamination in general. Okay, so how are bacteria influencing the lagoon? How could bacteria possibly be influencing health of the population around the lagoon? The other side grew out of some of the work with the Marine Mammal Group, which is looking at antibiotic resistant bacteria in the Indian Lagoon. Okay, so I'll be talking about each of these projects separately and maybe even keep on time. Okay, Dennis showed this graph. These are the long-term monitoring sites that he's been using for the last 10 years or more throughout the Indian River Lagoon, ranging from the Fort Pierce Inlet up through Vero Beach. And so we can see areas here which are influenced by input from the canal systems from urban development and compare those to the areas down near the inlet where we have better water exchange, less obvious contamination sources, okay? So we can refer to the top 3 sites as more impacted, the lower 3 sites as potentially more healthy. And so we can start to look at these sites and see what sort of things are going on. What I am interested in is microbial water quality. So looking at indicator organisms. Indicator organisms are a set of bacteria which are typically used to work out how safe water is, be that drinking water, recreational water, wastewater, okay? They are the tests which are done for things called fecal indicators. And these are bacteria which are commonly found in association with fecal material. So there are several of these groups. Down the bottom here we can see coliforms. That includes Escherichia coli and other thermotolerant coliforms, which are present in all fecal materials. Also the enterococci, okay? So these are present in very large numbers in fecal material. If you find these in a water body, it implies that there is a source of contamination going on. These are very standard bacteriological methods, and they allow universal comparison of data sets. So the first thing we did a few years ago was to set up these assays and see what was going on. These are the assays which give the indication to the local governments that the water is not safe to enter. So when you see this sort of sign showing up, these are both from down in Martin County, saying high bacterial levels in the water, these are the sort of tests they've done to be able to determine that you shouldn't be in there. And the state of Florida issues standards, okay? So for fecal coliforms, if you've got less than 199 CFUs per 100 mils, it's good water quality. A CFU is a colony forming unit. So when you take a water sample and you put it onto a petri dish, if you get bacteria growing, they form a colony. And one colony is one colony forming unit, okay? So if you've got a plate which has 199 colonies on it, it's good. If it has 200, it's moderate, okay? If it's above 400, it is poor water quality. For enterococci, the numbers are far lower. Good is below 35, poor is over 105, okay? But we can do these assays and get some sort of idea as to what sort of things are going on in the Indian River. So we have been doing these tests for the last, it's close to 3 years. We've nearly got a 3-year cycle on this data. And each time I present this data, I'm trying different ways to make it interpretable. I'm hoping that this works, okay? So, they're small graphs, but they're all there. So these are the 6 sites, and we have been monitoring these sites on a monthly basis for nearly 3 years, okay? So you can see from this end, this is the northern extreme down to the southern extreme. So from North Grand Harbor down to Torpy Road. I took all the data, I pooled it into 3 monthly batches. So January through March, April through June, July through September, and then October through December, okay? And you can see clearly that, firstly, there is huge variability, okay? You look at the error bars on these charts, they are huge. That's because you're comparing year to year to year. You get lots of variability between different years, okay? But you do certainly see trends going on. So the wet season, the rainy season, once you get into the summer, you see higher numbers, okay? So that's true in the northern area here. As you get down towards the inlet, that seasonality is not as obvious, but there is still a low number in the very dry season, okay? So we certainly have some influence going on with seasonality throughout the lagoon. If we look at the enterococci, we do not see the same level of seasonality, but what we do see is a distinct decrease as we go from north down to south. So the levels of enterococci in the southernmost sites are very low. We're also monitoring what are called heterotrophs. And heterotrophs is just an indicator of the total number of bacteria in the water. And again, you can see that there is an influence from rainfall, from the rainy season. The numbers of heterotrophs are far higher than they are in the dry season. And as we go down south, the variability decreases quite considerably. I had to put all these things on different scales because if you look at the number of CFUs here, we're dealing with the scale from 0 up to several thousand. Here we're in the hundreds, and here we're in the millions. Okay, so you can't put all these things on the same chart. Or if we refer back to the state standards, over 400 is poor. The 400 mark is right in here. Okay, so pretty much all the time, according to coliforms, we're dealing with poor water quality. With enterococci, we're dealing with generally fairly good water quality. So what is going on with this? If we're trying to work out what is really going on with human activity in these samples, but we have to remember that all these indicator organisms are just coming from fecal material. They could be coming from wildlife, from agricultural activities, from other sources. So what we've been doing recently is developing a human-specific test using Bacteroides HF183. To save time, I'm simply saying that come to the talk next Thursday. Mengling Zhao, my intern, has been doing this work, and she'll be presenting the work on the human indicator. We also would like to get some work going with the cow and bird to get indicators of agricultural and wildlife fecal inputs into these systems. The other side of the work that we've been doing is antibiotic resistant bacterial testing. Last time I spoke, we've been testing using plate counting. So just taking samples of sediment, putting them onto a medium which contains an antibiotic and seeing how many things grow. We saw huge changes in the numbers of resistant bacteria, which seemed to be increasing annually. What we've done now is developed a new method in collaboration with Dr. Esiubu down at the Davie campus. She's doing a lot of environmental testing, which this allows us to test a larger number of antibiotics. It's far less labor intensive. And the way we do this is we take a sediment sample, we put it into our broth. Anything which will grow in the broth, we can then plate out onto an agar plate. We can then apply discs with antibiotics on them, see if we have antibiotic sensitivity. This is a method which is typically used in medical microbiology. So we can test a whole series of antibiotics, and we can determine whether they are actually sensitive to that antibiotic, intermediate, or resistant. bring your attention to 3 of these: piperacillin, SXT, which you probably know better as Bacterium, and the last one there is vancomycin. And this then goes through a time series from August of last year through the present. You can see we start off with red is resistant. This is intermediate. Here we have intermediate and sensitive. The vancomycin is resistant, okay? And just keep watching those lines there, and you will see things start to change. Piperacillin resistance is increasing, okay? And we have total resistance to Bacterium over this time period, okay? Vancomycin, the samples are always resistant. Okay, so we have bacterial resistance to many classes of antibiotics in the lagoon. We have resistance to vancomycin. Vancomycin is the antibiotic of last resort, but we're seeing resistance all over the place, and we're seeing variable levels of resistance to piperacillin and Bacterium. Okay, thank everybody in the lab. Thank Adam, who's the co-PI on this, my interns, and Dennis, without whom we wouldn't have samples to test. Thank you.